Thank you for joining me today. The pandemic helped change the speed of adoption and use of our C1 gene expression platform. We and a growing number of scientists worldwide believe that Dyadic's robust and hyper-productive C1 vaccine and drug manufacturing platform is an ideal solution to speed of development and produce massive quantities of vaccines quickly and at much lower cost at flexible commercial scales. We at Dyadic, along with a number of our collaborators, are committed to helping bring health equity to patients globally. I want to remind you to review our prior filings, including our risk factors, as well as information on our website and other public disclosures, as information related to our science and our business is constantly changing, which may be material to investors. Our C1 technology has and continues to demonstrate the versatility to efficiently speed the development and enable the production of very large quantities of more affordable vaccines, antibodies, enzymes, and other protein-based products for use in a growing number of applications across human and animal health, including record levels of antigen vaccine production, as shown in the data presented by world-renowned animal and human health infectious disease scientists during the Zoonosis Anticipation Preparative Initiative, the ZAPI Stakeholders Final Conference held February 4th and 5th, 2001. The world and governments are focused more now than ever on global health equity. In order to achieve health equity globally, there needs to be disruption in the way biologic vaccines and drugs are developed and manufactured. We at Dyadic, along with our government, academic, and industry collaborators, are developing what we believe is a highly disruptive biologics manufacturing platform that is beginning to demonstrate the potential to revolutionize the way animal and human health biologics are developed and produced. We have a solid financial position to continue leveraging our C1 technology for its use across a growing number of applications in animal and human health, including expanding our infectious disease franchise, both for prevention, such as recombinant protein vaccines, and treatments such as antibodies, and for other inflammatory, metabolic, neoplastic, degenerative, and other diseases, including COVID-19, to allow for fair and equitable global access. Our addressable markets are enormous. The C1 technology platform can be used for these applications and many others, including growth factors, as you noticed recently in an announcement with Turtle Tree Scientific, where we're working on developing growth factors to be produced potentially from our C1 technology. We're building on our 2020 success in 2021. The business development and the science momentum are rapidly changing and moving in the right direction. We're moving towards our first potential phase 1 clinical study with a C1 RBD antigen against the COVID-19 pandemic. We're leveraging the success in developing C1 antigens against SARS-CoV-2 and a variety of animal viruses for ZAPI to expand the recombinant vaccine pipeline for human and animal health. We're continuing to strengthen and expand scientific and business leadership capabilities to execute world-class business development and leverage strategic collaborations for C1-based biologics. As we advance our science, we continue to expand its use. We advance our own pipeline as well as those of our collaborators, and we're moving into cGMP manufacturing to produce tox and clinical trial grade material as we move towards initiating further internal and external animal and human health clinical trials. Our focus for 2021 starts out with continuing to prepare for our potential first phase 1 human trial using our Dyadic-owned C1 cell manufactured SARS-CoV-2 receptor binding domain, RBD, recombinant antigen vaccine product candidate. The RBD from C1 is going to be used as it would be believed to be a higher neutralizing antibody and a potentially safer vaccine candidate than the full spike proteins from the warp speed candidates that are out on the market today and that are coming soon. We're looking to expand the RBD antigens to cover other areas, South African, Brazilian, potentially New York, Californian, and the changing variants, as we anticipate the first phase 1 human clinical trial, either in the EU, the US, or otherwise, with a C1 cell manufactured SARS-CoV-2 monoclonal antibody. We're continuing to advance commercial recombinant antigen vaccine product candidates in animal trials with global animal health companies. And we're generating additional safety and efficacy data in addition to that which was demonstrated recently by ZAPI. They're also working now on additional trials for both SPV and RVFV recombinant antigen vaccines in 2021 to further demonstrate the safety and efficacy in addition to the hyperproductivity. On the human biopharmaceutical programs, we continue to advance immunotherapeutic monoclonal antibodies with top-tier biopharmaceutical companies, including with a top-tier biopharmaceutical company with registrational potential in oncology. We're continuing to advance immunotherapeutic bispecific monoclonal antibodies with a variety of pharmaceutical companies, including a top-tier biopharmaceutical company with registrational potential in autoimmune disease. We're advancing our glycoengineering efforts in our Nivolumab, or the Opdivo potential biosimilar, is our monoclonal antibody product candidate proof of concept molecule. As you know, the European Union's Zoonosis Anticipation Preparedness Initiative, ZAPI Stakeholders Final Conference, was just a few weeks ago. In that conference, they repeatedly referenced Dyadic and our C1 gene expression platform. They demonstrated in the ZAPI program over the 5 years, Dyadic played a key role in that program. Within the collaboration, we helped to validate a prolific fungus developed by one of our project partners, Dyadic, who demonstrated their C1 technology. This was a statement by Dr. Jean-Christophe Audonnet, who works for Boehringer Ingelheim and was the project coordinator from ZAPI. You can see here, as he comments, that the C1 cell line far exceeded their initial expectations, and we turned in record antigen productivity for both the SPV and RVFV antigens. Information about ZAPI and the stakeholders' final conference are now available on the IABS website, and recordings are available on YouTube at the links below. I'd like to play for you a few of the highlights and conversations from several of the scientists within the ZAPI program. This particular slide is going to be a presentation and just quick brief comments by Dr. Albert Osterhaus of Erasmus Medical Center, who is also a co-manager of the ZAPI program. So I'll play for you right now that slide.
The manufacturing processes here, there's the efficient manufacturing processes are being developed. I specifically would like to mention Dyadic, the C1, the fungal process there, which gives enormous amounts of antigens. And I think, especially in the face of the world situation with the pandemic, a system where you can produce much more vaccine would be an ideal one to look into. So if that is if that were to be the only spin-off, that would already be fantastic. The manufacturing process.
Another statement made by another person, Dr. Alexander Brix, another leading scientist, who in this case worked for Boehringer Ingelheim, a major pharmaceutical company. And here's what he had to say about the C1 technology platform.
However, if we look at the subunit production that was just also described by Ronan, it is pretty clear that the C1 expression or using the C1 platform is the key advantage here for, as it's described, for yields, but also DSP, purification. We don't have to compare those numbers again, but as Ronan described, there's grams per liter base yields, and in terms of surge capacity, that's what we need. And therefore, for further development, C1 now is or has been chosen as the production house for those subunits. Fast development, and I repeat again, sufficient amounts. And what's also very important then from an industry perspective is the reduced fermentation volumes. We don't need 4, 5, 6,000 liters. We maybe need 10, 50, 100. And we can use smaller systems and more flexible systems, obviously. And I think as it's written here, that's exactly what we want to have, and it's exactly in line with the ZAPI objectives.
In addition to Dr. Osterhaus and Dr. Brix, there are a number of other scientists that make presentations that refer to the power, the speed, the safety, and efficacy of the antigens produced from the C1 technology. However, if you look at the success that we had in ZAPI is revolutionary. They mentioned during the presentations recently that it was, if nothing else, using C1 and that technology and the C1 expression levels, the safety, the efficacy, its breadth, its scope, its diversity of use provides for a phenomenal success for ZAPI and for Dyadic. We've gone a long way in developing the technology, using the technology that we've developed on our own and in conjunction with ZAPI and others over the last several years during that program. We demonstrated in that program 300 times higher productivity than the insect cells used in the program for the SPV antigen or baculovirus. Same thing for the Rift Valley fever virus. And of course, our C1 cell technology not only produces massive amounts of low-cost proteins, it does so at flexible commercial scales using standard microbial fermenters that are available virtually in every country across the globe. In addition, during this time frame, starting in 2018, 3 years later, we started a collaboration with the Israel Institute for Biological Research, or the IIBR. In this collaboration, C1 demonstrated that we could produce not only an enzyme to protect potential attacks from sarin and nerve gas and other VX gas attacks that could potentially are biological threats, but we embarked upon a journey to develop another SARS-CoV-2 receptor binding domain vaccine candidate. The Israelis demonstrated that they can produce that in very large quantities at low cost, which neutralizes the COVID-19 virus. In addition, you'll see as we come up here that technology continues to be improved. The IIBR conducted a mice trial that demonstrated that the C1 cell produced a SARS-CoV-2 antigen that generated high levels of antibody binding and neutralization. And they're completing and studying and coming up with the analysis of their IBR transgenic mice human ACE2 challenge data, which is expected very soon. One of the things that we've developed within this last year was to develop a method of how to go from a gene sequence into creating a stable cell line, to doing animal studies, to then producing materials for tox studies, and then for clinical trials. And then we have joined in the support of not only animal health trials and collaborations, not only CGMP manufacturing collaborations and collaborators, but the support for regulatory and CMC consultation with Parexel, who is one of the leading CMC and regulatory advisors all over the world. In addition, CR2O is coordinating the clinical trials of the expected upcoming phase 1 clinical trial for our SARS-CoV-2 RBD vaccine. We can repeat this time and time again, potentially with the South African variant, the Brazilian variant, potentially a California variant, a New York variant, or any variant that us and or our collaborators or governmental agencies would like us to try. We can make potentially trivalent and tetravalent vaccines at levels that I don't think anyone else can, at the cost and the quantities that the global world needs to help fight the pandemic. In this particular case, as we just mentioned, we can produce stable cell lines in approximately 2 months or less. We take the gene sequence, We apply it. We create stable cell lines very rapidly that produce very, very high levels of antigens. We then can move those forward. And we've done that, as we mentioned, with the SARS-CoV-2 as the proof of concept and the RBD. And now we're working on, as I just mentioned, the potential to do that with up to 4 critical variant RBDs and potentially 1 or more of the full spike proteins or other varieties of vaccine candidates that we're evaluating and being offered by numerous scientific collaborators around the globe. In addition, we're working on monovalent and potential multivalent monoclonal antibodies for SARS-CoV-2 product candidates and otherwise. We've executed an MTA with a top-tier pharmaceutical company to perform a fully funded research and evaluation of a C1-produced SARS-CoV-2 monoclonal antibody. We have already transferred that material to that pharmaceutical company, who's beginning to do the evaluation and analysis of the quantities and the qualities and how that stacks up against the CHO-produced monoclonal antibodies. We anticipate potentially entering into a collaboration with the former ZAPI EU scientists and other parties to express 1 or more additional SARS-CoV-2 monoclonal antibodies with the objective of leading to an additional phase 1 human clinical trial of a single mAb or a cocktail mAb. Sorry about that. Expressed from the C1 cells. So the key here is we can transfer this technology very rapidly. Again, as we mentioned, and I've mentioned earlier, we use standard either single-use bioreactors or stainless steel tanks to produce proteins and biologics from C1. Those cGMP microbial fermenters are available on a global basis. As we mentioned in prior years, 2020, we conducted a tech transfer of C1 into a number of organizations, including CDMOs and pharmaceutical companies, including WuXi Biologics, and now also, as mentioned earlier, Biotechnology General in Israel. In 2021, we're completing our first engineering run, and we've initiated our second engineering run and have just successfully completed that. Our objective is to produce the SARS-CoV-2 recombinant antigen under cGMP in support of our tox studies and the potential phase 1 clinical trial. In addition, we're working with Oslo University on an influenza and potential are evaluating from both the NIH and the ZAPI group and others, potential other infectious disease pathogens, either as vaccines and or as antibodies. We mentioned a few weeks ago, we entered into a collaboration with Turtle Tree Scientific to try and produce a number of growth factors, which could be used for regenerative medicine and other purposes. And again, fully funded proof of concept protein expression programs are not only helping us move those programs forward, but we're learning more, expanding the use, the breadth, and the scope of the areas of which we can apply the C1 platform technology for ourselves and for our collaborators. One of the products that we're working with is on our own is Nivolumab. It's our model protein to try to make monoclonal antibodies with the proper glycoengineering or the human-like glycan forms. This proof of concept is successful, will lead to a potentially huge opportunity, not only for us to potentially out-license or collaborate with or partner this particular cell line to produce Nivolumab, but it sets us up for success in following on with multiple other candidates for monoclonal antibodies or other programs like bispecifics, other antibodies that need human-like glycosylation. This is a huge opportunity and one that we're pursuing as our proof of concept glycoengineering program, which we've successfully engineered C1 cells now to produce G0 and G2, G1 structures. So now we'll talk about the competitive advantages and why people are so excited. So C1, as we mentioned, is a filamentous fungal cell. However, it grows very unusually, not like typical filamentous fungal cells. The morphology, the physical form, the structure allows for hyperproductivity, 100-folds different than the beginning cell line under lower viscosity, which means scalability. As you know, in biotech, there's a saying, fail to scale. Well, we've scaled in the industrial biotech world up to 500,000 liters. In the pharmaceutical industry, the largest vessels that people are using typically are up to 20,000 liters, or 1/25th the size we've already scaled C1. So we're scaling down. So fail to scale, I don't think is going to be relevant here and doesn't look to be relevant in our existing programs. We have proven low cost, high yield, purity, and scalability, transferability. And so what we believe here is we can bring broad benefits to a global population, not only for pandemics, but also for traditional vaccines and drugs that are used in our daily life. The C1 cell recombinant protein products have competitive advantages in terms of purities, productivities, robustness, speeds, and cost. Yet again, the world is now facing dilemmas. How do we get global health equity? And the answer is we need a more vigorous or vicious, robust cell line to produce quicker, faster, cheaper biologics that can be made more affordably and can be tech transferred locally so that we can have better distribution, lower cost, and more affordable, more accessible healthcare to patients globally. And we believe, and a growing number of people believe, and scientists, that C1 has the potential to disrupt biomanufacturing and deliver on that promise. This is an example that shows you why we think we can deliver on that promise and why other people believe that we can deliver on that promise. Giving an example, like in ZAPI or in our SARS-CoV-2 RBD program, if you look at the middle on antigens, we've produced the RBD at up to 3,500 milligrams per liter. As I mentioned, in the ZAPI program using insect cells or baculovirus with the SPV antigen, the nearest competitor, the insect cells or baculovirus, was 6 micrograms per liter. In that program, we were 1,800 micrograms per liter where we left off. That was 300 times greater. We saw even a bigger difference between the Rift Valley fever virus antigen, between the insect cells or baculovirus cells and C1. Monoclonal antibodies in the non-glycoengineered cells, we've seen up to 24.5 grams per liter or 3.1 grams per liter per day. And that compares with a typical CHO cell process that may get up to 10 or plus grams per liter in 14 days. So there's a huge difference. The media cost is de minimis compared to the CHO cell media. And then the FC fusion yields, the trispecific yields, the virus-like particle yields, the influenza yields. It looks like C1 has the potential to really make record levels of productivity across diverse multiple classes of proteins for diverse disease applications, both in animal and human health. And we're going to continue to pursue and push productivity levels to even higher levels in collaboration with our partners and on our own. So we have a focused business development strategy. It allows Dyadic to develop and advance the C1 technology at a lower cost to the company because the funded proof of concept collaborations for specific therapeutic products have helped us identify which genes to remove to reduce the proteases, which antibodies, which classes, which drugs seem to be easier expressed in C1 and which ones aren't, and how to fix that and how to modify that and how to engineer C1 cells to be able to produce higher, greater numbers of more stable with antibodies, therapeutic drugs, and vaccines with the right properties and the right characteristics. We believe that's going to lead us to upfront access fees, milestones, and royalty payments, just as we saw in the industrial biotech with the industrial business, which we licensed technologies to BASF, Shell Oil, Amigo Bioenergy, and then DuPont purchased our industrial business at December 31st, 2015 for $75 million. Small and medium biotechs, potential to get equity in milestones and royalty payments. Here, what we're working on with small biotechs is they can't afford to provide us with upfront fees because they need that money to advance their own biologic programs. So we're taking equity in some of those biotechs, fully funded research in most of them, to get more milestones and more royalty payments on more candidates, to build a portfolio of opportunities of where we can collect revenues. And then grants and contracts, just as the IIBR and the ZAPI group, which led to a lot of investments to help us advance and develop the C1 technology to generate data that demonstrates the revolutionary potential and the ability to produce large amounts of low-cost proteins that have efficacy and safety, which we've demonstrated in ZAPI and other programs. And then we have our internal dyadic product development. We've talked about in the glycoengineering, where we're advancing Nivolumab as a proof of concept for production of monoclonal antibodies as a class, but we also can use that to out-license it as a particular product opportunity. We've deleted proteases and done other engineering to the C1 cell line. That's led to hyperproductivity of not only the RVFV and the SPV, but also the SARS-CoV-2 antigens and antibodies. We're working on influenza, and we're looking at other infectious disease and certain metabolites. Again, we have a solid financial position as of Q3 with $30.5 million in cash and investment securities, no debt, no warrants. We have partnerships with top global partners and our wholly owned programs. We have robust scientific data. We've demonstrated successful productivity at record levels, but also safety and efficacy in many of these programs. Our emphasis is on large and growing addressable global human animal health markets with many shots on goal and more to come. We've well-established global biological R&D organizations, top-tier human and animal health pharmaceutical companies, as well as governmental and private agencies. And we have a highly experienced and energized professional management team and a world-class board of directors. This slide indicates our management team. We'll start with Matt Jones, who is our business development leader, CCO. Matt worked at Lonza and GSK. Ronan Chelet, Dr. Chelet's our CSO. He worked at Teva. He worked at Codexis. While he was at Codexis for multiple years, he worked on C1 in the Shell program, where several hundred millions of dollars were spent in the Shell program with C1. And then Ping Rawson, our CFO, has had 20 plus years of finance and accounting. And of course, I'm the founder and CEO, and have engineered the success of C1 and Dyadic over the last several decades. On the board of directors, I believe we have a world-class focused board of directors that are very active. To start with on the left, Dr. Aaron Bose, 34 years bioprocess development at Pfizer. He's also a National Academy of Engineer member. Dr. Buckland, same kind of pedigree, but more on vaccines, where Dr. Bose is more on the antibodies. On the vaccines, Dr. Buckland worked at Merck, 29 years of R&D leadership. He's also a U.S. National Academy of Engineer member. Michael Tarnock, chairman of the board. He also worked at Pfizer for over 3 decades and also at Carex. Patrick Lucy, our newest member, worked at Phoenix. And as you might recall, Phoenix recently just sold their business to Ligand for over $500 million in valuation for shareholders. And also, Phoenix was a microbial cell line expression system company using a prokaryotic cell line, which could produce non-glycoproteins. And again, we believe C1 has broader, deeper, wider use and applications because we can make glycoproteins as well as non-glycoproteins. There's a global need, and it's growing and it's rapid, for safe, scalable vaccine, antigen, and antibody and other drug manufacturing. Unmet needs still exist for SARS-CoV-2. Although we have now Pfizer, Moderna, and recently J&J, there's a need for larger volumes of lower cost variant vaccines, booster vaccines, a variety of maybe potentially better vaccines. The aims and milestones that we have set for ourselves are high. We're combating infectious disease, inflammatory, metabolic, neoplastic, degenerative, and other disease, including COVID-19, but not limited to that. Our focus is on human and animal health in traditional drug development and production to allow for fair and equitable global access. Strategies is to apply this very, very powerful production system to produce a variety of diverse and growing needs and fulfill those needs of biologic proteins, such as vaccines and antibodies, to deliver the world a low-cost solution for biologic drugs and vaccines to a global population. As you can see here, we've been successful in working for preclinical programs in many countries across the globe. We're focused now on a few of those programs, and in particular on our SARS-CoV-2 RBD program, moving that towards toxicology studies and a potential phase 1 clinical trial this year with our own SARS-CoV-2 vaccine. In summary, we want to accelerate the adoption and use of C1 cells to speed the development, lower the cost, and improve the performance while providing the necessary quantities of vaccines and drugs to a global population more affordably. The strategy is to deliver these cells to multiple parties all over the globe, to use it for ourselves, to develop our own products, to use it in collaboration with others, in partnerships, where we get milestones and royalties and upfront access fees. To expand our infectious disease franchise to include bacterial, fungal, and parasitic pathogens. The potential opportunity to advance immunological vaccines, antibody, and other candidates into human clinical trials is almost unlimited. To extend C1, the recombinant glycoprotein manufacturing technology, into new diseases, including autoimmunity, degeneration, inflammation, metabolism, oncology, and others. To expand our footprint globally and to expand in the countries we're in, including into North and South America, Europe, Africa, Asia, and India. To continue to build highly experienced, intelligent, creative, ethical professionals to help us grow our business and to expand the opportunities of where we can apply the C1 technology for the benefit of humankind. And to continue to build a reputation for global manufacturing, business development, strategic partnership, and commercialization of Dyadic franchise products, including with potential Dyadic spin-out companies, partnerships, or other types of collaborations. I want to thank you for joining me today at Dyadic, and feel free to contact me at the above email or at our website, www.dyadic.com. Thank you.